webinar in the series on IMMray PanCan-d. Today we will discuss the commercial test model study and give an update on all our launch activities. Today our presenters will be Dr. Linda Mellby, Vice President of Research and Development, Dr. Laura Chirica, Chief Commercial Officer, and Dr. Tom King, our Medical Director. Our agenda will begin with a discussion by Linda on our IMMray technology overview. Then it'll be followed by the commercial test model study results. And then Laura will give an update on our US launch program and the status. And finally, we will discuss the final clinical validation stages for the launch for Q4 2020 with Dr. Tom King. After that, we will have a question and answer session, and we urge you to type in your questions, and we will be moderating those questions as we get to that section in our webinar. We are only taking typed questions today, so please log in and type in your questions. With that, I will turn the call over to Linda to give us our overview.
Yes, thank you, Judy. So I will start with the short overview of Immunovia, the company. So Immunovia was founded 2007 from Translational Cancer Center at Lund University, the largest university in Scandinavia. We then took on several years of research and development of the IMMray technology and went into a product development of our pancreatic cancer test, IMMray PanCan-d. And this is our first product for a major clinical need. And we are now in the last steps in the development process, entering the commercialization phase, and we will provide commercial testing on our Immunovia DX laboratories situated in Boston and in Lund. So during this webinar, we will go through some major study results that are very important for us, as well as the coming and ongoing launch activities to prepare for the launch of our pancreatic cancer test. So before doing that, I will just give a short overview of the IMMray technology. So the IMMray technology provides us opportunities to develop antibody signatures and biomarker signatures. And biomarker signatures are combinations of biomarkers that could separate, for example, pancreatic cancer samples and patients versus controls. And we are developing these signatures by measuring both immunoregulatory proteins and tumor cancer-associated proteins. And we are using antibodies as catcher molecules to measure these proteins in blood. These antibodies are printed onto a slide, and that is an array, antibody microarray. And we then add pre-labeled serum samples to these arrays, and the bind targets analytes are creating signal patterns that are interpreted by our bioinformatic algorithms, and biomarker signatures for unmet clinical needs could be generated. So these antibody microarrays with the signatures are produced in-house in Immunovia at Immunovia in Lund in Sweden, and we then ship these to our Immunovia DX laboratories for patient testing. So in the DX laboratories, they receive patient samples, and only a droplet of blood is needed. These are analyzed on the antibody microarrays, and a test answer is calculated in a cloud-based software where we have all the algorithms, bioinformatic algorithms and calculations. And then a test report with actionable results for clinicians are a result as coming back from the software. So this is the overview I would like to give you of the technology platform, and we are now using this for the development of our pancreatic cancer test. So during the development of IMMray PanCan-d, we have run several discovery and development studies, and the latest was the IMMray PanCan-d commercial test model study that we finalized end of last year. So this is a study that I will tell you more about right now. So as I said, we have run through several studies during the development process. And the first one, we showed that we could detect early stages of pancreatic cancer, meaning stage 1 and 2 versus healthy controls. And that was a major finding, and the studies were published in Journal of Clinical Oncology 2018. And then during the continuation of the development process, we have adapted more and more to a real clinical situation where the symptomatic controls become very important and become a very important clinical intended use population. So in the commercial test model study, we show that we can separate early pancreatic cancer stage 1 and 2 versus these symptomatic controls. And in this process, we needed to fine-tune and adjust the biomarker signature towards these types of controls. So the commercial test model study was a multi-center case control study. We collected prospectively from several sites in Europe and US. And in this way, we tried to mimic a real clinical situation already in this stage in the development process. And that is very important because we have very high quality samples that we use to fine tune and lock our commercial IMMray PanCan-d biomarker signature. And we have then finalized our PanCan-d test model using this signature and locked all the bioinformatic algorithms. We have set the product configuration, all the quality control parameters, and so on. We also showed the predicted performance of this test model using these very clinically relevant control groups. So in total, we had over 1,100 serum samples, whereof 350 PDAC all stages. About 1/3 of these were early stage pancreatic cancer samples. We had 310 healthy controls and 488 symptomatic controls. And these were all collected from 8 different reference cancer sites that we have in collaboration with our leading key opinion leader network. These were collected in London, in UK, and in the US, in Spain, Sweden. And by having these samples collected from all these different sites, we increase the biological availability, the analytic availability, and also the pre-analytic availability when it comes to sample collection, storage handling, and so on. And this, in this way, stressing the system made us proving the robustness of the PanCan-d test model and the selected signature. So another very important aspect of this is to have clinical relevant controls that reflects the clinical intended use of the test. And in this study, we had a large number of non-PDAC controls being prospectively collected at secondary care centers and gastrointestinal units. These are patients coming in with symptoms suggestive of pancreatic cancer, but not being pancreatic cancer. Instead, they are diagnosed with various benign pancreatic and biliary diseases, such as chronic pancreatitis, acute pancreatitis, pancreatic cyst, liver diseases, and also diabetes. So with regard to the positioning of the PanCan-d test, this is really a spot-on clinical intended use population. Worth mentioning also in this context is that all these samples were fully randomized, both on primary diagnosis, but also on other clinical relevant parameters over slides and days of analysis to reduce any technical biases along the run of the study. So our IMMray PanCan-d test model was built upon using all these pancreatic cancer samples, 315 in total, versus all non-PDAC controls, 798 in total. And one major finding in the study was that we could reduce our PanCan-d biomarker signature to a very focused and robust signature, including only 8 biomarkers plus C99. So we used this biomarker to build the model and then to generate performance data we divided this data into training and test set. And the results I will present now, it's only from the test set, and that is very important to not have any overfitting or other biases during the data analysis. So the classification performance of the IMMray PanCan-d using the 8-plex biomarker signature together with C99 show that we could separate all pancreatic cancer cases versus all the controls combined with a ROC-OVC value of 0.94. And focusing only on the symptomatic control group versus all PDAC, we had a ROC-OVC value of 0.93. So continuing with only looking at the healthy controls, we could separate these versus all PDACs with a ROC-OVC value of 0.96, a slight better as expected. And when we focus on only the early stages of PDAC, stage 1 and 2, versus all the controls, we had a ROC-OVC value of 0.95. So we were very pleased to see that we have the same classification potential for the early stages of PDAC as for all PDACs, and also that we have this high classification performance using the 8-plex biomarker signature together with C99. So the major outcomes of this study was that we have an excellent predictive performance of the IMMray PanCan-d test model in separating pancreatic cancer versus all these clinical relevant controls, including the symptomatic controls in a multi-center setting in a study that we run multi-center, have collected samples from several sites. Another major achievement is that we have refined our signature down to a 8-plex biomarker signature together with C99. And from a product perspective, there are many advantages with reducing the assay complexity, the technical availability, the costs, with facilitating our production processes as well as QRA processes. So there are many advantages with reducing and still keeping the performance of the signature. We have locked it, the commercial signature. We have filed the patent in July this year. So now we have a 20-year protection from this date. So with these excellent results, we have now entered into verification and validation activities. And the clinical verification study is ongoing. In this study, we have also prospectively collected independent sample cohorts from several sites in Europe and US. These will be run, of course, on a locked PanCan-d model. We will also have locked production processes, product configuration, software design. We have done a tremendous work in setting up all the hundreds of specifications and work instructions to have everything in place. And now we are verifying all the production processes and the design, and the clinical verification study is an important part of that. We will also verify, of course, the clinical performance of the test model when classifying pancreatic cancer patients from controls. And we will finalize this study end of Q3 this year. And the next step is the final clinical validation that will lead to cell start Q4 this year. And Thomas King will talk more about this and the other validation activities that are needed for the launch in US. But before that, I would like to leave the word over to Laura that will give an update on the launch program and all the activities leading to the preparation for the US launch. Thank you very much, and Laura.
So, hello, everybody. And it's always a very interesting experience that we are now going through all these virtual conferences. And I have to say that I miss having the audience close to us, and we are looking forward to getting back to having the conferences and the face-to-face meetings in which you get the contact with the audience. But nevertheless, I know that you are out there, and I have to thank you for joining us and for taking the time to listen to us. I am going to take you through the US launch program. I'm in charge of commercialization of Immunovia's products, and I'm, let's say, much more excited because now we are going to introduce to the market our first product. And as you have heard from Linda, we have fantastic results. We have an amazing product which has the potential to revolutionize the way the pancreatic cancer is detected and diagnosed. And in this way, we also have the potential to detect the cancer early enough for the operation, which in its turn is going to contribute to much better survival rates for the patients. 5-year survival rate today is about, let's say, an average 9%, which detecting it early leads to up to 50% 5-year survival rate. So a fantastic, significant improvement for the patients and their families. Because of that, it is actually an amazing journey that we have been through. And I want to give you a context before I'm going into the launch itself. And for that, I just want to take you through, in just a few words, the road to market. We started already 2015 with establishing the very important key opinion leader collaborations, and we managed today to have in the US only 15 ongoing collaborations and studies in the prospective study, of course, and also we are now working on all kinds of different studies that are needed for market expansion, and these are also extremely crucial, as I'm going to discuss further on in the presentation, for the different types of insurance coverage, and of course in the end to achieve reimbursement and inclusion in the national guidelines. So now let's have a look to the different programs that are included into a launch that should be extremely successful. And it is a number of different processes that are all, let's say, going in parallel, and they are all contributing to let's call it a commercial readiness, and also to contributing to having all the target groups on board and ready to use, order, and have the test in their hands. We have here the 7 programs, and as you heard from Linda, Dr. King is going to take us through the laboratory processes that have been successfully achieved in Marlborough, and the work on the regulatory approval, and he's going to actually do it after I take you through the other remaining programs. As I said, clinical studies play a very important role. Actually, I would say, coming with the background in diagnosis in different types of cancer and autoimmune diseases, I would say that for us, clinical studies here in Immunovia are even more important because there is not any other test on the market. So we are the first. And we are working with now 2 types of studies. We have in here the market expansion studies, which are very important to be performed with US patients, so US samples in this case, that are also going to work on the intended use cohorts, and that are going to be used together with all the results are going to be put together into the reimbursement test portfolio that is used to submit for different kind of coverage and insurance. Then we have been also working with the 3 prospective sites, and we are covering in total in these multi-center studies more than 10,000 risk individuals, with the 15 sites only covering American patients. So this is extremely tightly connected into the reimbursement program. And this program has also been going on for quite some time, part of the market access activities that we have been performing during these years. And it starts with a lot of payer discussions and payer interaction and meetings in which we have been defining their needs and how the test portfolio should need to look like and what kind of information should contain in terms of the clinical data, in terms of the health economy data, and all of these have been included in the design of our studies. And with this, we also defined how the self-pay model is going to work until we're going to achieve reimbursement. Then this payer engagement is, of course, continuing, and also there the prospective study play a very important role in delivering the results that will then, with their final readout, lead to the reimbursed sales. And in the meantime, the market expansion studies will provide all the results needed. If we then switch gears and we now have a look at a very important operational process that is part of any successful launch, is to have the logistics in place. And we have been working with defining all the steps in the logistics. Of course, US is a large country and with quite a lot of distance to be covered. So we had to find a solution, logistic solution that is convenient, especially for the patient, for the risk group individuals, and also for the clinician to order. I will just lift two of these steps here. One is the fact that our test requisition, test referral, has been developed so that it contains a lot of supporting material that is not only for the clinician, but also for the patient, because we have an extremely important communication and contact with the patient in this terrible disease. Then I want to lift another step in this logistics, which our American colleagues have really done a fantastic job. So I want to just applaud their creativity, where they found the possibility to have remote phlebotomy. When I heard it the first time, I just, of course, in Sweden, for example, we don't have that. We have similar solutions now, which have been taken to Europe, but at the time we started with it, it was really something totally new. Then we realized, and this is actually quite interesting, that during these strange times that we are living the last year, remote phlebotomy became actually the method of choice, because patients don't want to go and enter the hospitals. So the phlebotomists are going to the convenient location for the patient. They are programmed to meet them, and they have all the equipment needed to protect themselves and the patient. So we have tested this logistics solution for our prospective collection already, and it works extremely fine. Then I will also add to this picture what we all know is so important, the customer support. We all know how much we need customer support, and especially when we have such a new innovative solution like ours. So also here we have created customer support processes that are unique for us and for our test. And we have a combination of a traditional support that is, of course, convenient, and it's also in a timely manner. But not only that, we also added, again, due to the nature of pancreatic cancer and due to the nature of this terrible disease, we have extended the support into a package that has an extended patient support, so that we are able to monitor each and every step in the test processes. And just make sure that we are able to answer and work through with the patients and the clinicians in a thoughtful way, in a compassionate way, and also in an urgent way. Then I will take you through the marketing and communication plan. Again, I think being in this field for more or less all my working life, and I am now passing the 20 years in this, I have to say that this has been the most amazing marketing and communication program I have been part with. And just the fact of being able to present our platform to the American Senate and to the health authorities and everything else that has followed just shows how much this is needed and what a fantastic test we are going to offer. And we are just there. So what we've done, looking at our primary message that is going to reach out to the patients, to the risk groups, and of course to our clinicians, is around the fact that early diagnosis saves lives. And in this context, we also were put in front of the situation this year. So we had to readjust and recreate our communication plan and making it a virtual one so that we are able to reach the risk groups and the clinicians through virtual channels. Here we are working with a number of different, let's call them tools. And in this case, in our toolbox, we have the virtual events. And in here, I just want to one more time bring to your attention our Immunovia Walk, or the iWalk campaign, which we have just recently introduced in July, August, and there we have an ambitious goal of reaching about 40,000 kilometers, which is actually the circumference of our planet, and doing that all together to raise awareness for pancreatic cancer. For the ones who are listening and have already contributed, we want to thank you. For the ones who are interested, we really need every step from everybody. Every kilometer counts, so that we can actually manage to get to our goal, and in this way engage as many people as possible in understanding this disease. You will find all the information through this presentation, and also just going onto our webpage on the banner on the homepage. Can start registering. Then we have been working on different kind of virtual events that are being provided instead of the classical ones that not everybody actually has turned yet into virtual. I think many will continue to do, and we see more and more doing that. But we already have confirmed to be part of primary care physician targeted event, virtual industry theater at PriMed conferences, in which we are going to have a live webinar. And also we are going to have a podcast series that is guaranteed to have quite a large audience each year. And also in the same, we are working with gastro and a gastro event, which also has been very important last year for us. So we are going to continue and have a virtual symposium there, again with the possibility to advertise in the same channel in which these gastroenterologists are always present, taking their courses, taking their information. Then I am going to just show you that we have also worked our virtual road tour. We have been talking about that, that we are going to reach our target clinicians in our prioritized hospitals, and instead of a lunch and learn, the kind of the classical event that we've been through in the early days with the bus that was traveling around. Now we are going to just have virtual ones and reach out to these people. We have also, of course, developed quite an extensive digital communication channel platform, and there we work on combination of online TV, which has become extremely popular these days, radio, online also, with local news. We are producing one segment that is going to be focused on pancreatic cancer with the Lifetime TV network that reaches 20 to 30 million Americans. And we're working also on different types of advertising that are both on the East Coast and also local ones. Then one very important part of the launch is, of course, having a campaign, and we have now developed our campaign for the launch, which is called the Imagine Early Detection, because it's been created in very close collaboration with patients, their families, and clinicians who were asked to answer the question, what does early detection mean for you? Early detection of pancreatic cancer, of course. So we're going to start releasing that. You see a preview here. And we are going to go through a 3-step rocket into all the different channels and advertisement. We equip our sales team that is now on board and working hard with the best possible tools now in digital format, and with e-marketing collaterals, with the possibility to do customer engagement tracking, which I think it's a big advantage for us. And last but not least, we have confirmed now to have on the launch day and also around the launch day 2 live stream digital events, one driven by Immunovia and the other part of the Primate conference, which takes place just around the launch time. So these are our marketing and communication activities. Before giving the word to Tom, I will give you an update on the last part of this programs, our sales program. In here, we have been focusing our efforts on clinical specialty, and as we have been talking many times, we have targeted the primary care physicians, or the general practitioners as they are called. The very important target group for us are the gastroenterologists, the pancreatic cancer surgeons, and also the genetic counselor who are assessing the familial hereditary risk individuals. Then we have also defined that our implementation and the way we are going to roll out our sales program is in the 4 territories on the East Coast, around, of course, our home market in around Boston and Massachusetts, and there we find these 4 territories. In this, we have actually already mapped all the different gastrointestinal centers, the multidisciplinary ones that are also having pancreatic diseases, and the different gastroenterologists and genetic counselor in those. Moving forward with this, we also looked at the pancreatic cancer centers in these areas and the National Pancreas Foundation accredited centers, which are extremely important in the processes that we just talked about, the reimbursement and different insurance processes. And then, for example, in here we see one of the territories in which we took and prioritized also those hospitals so that we have exactly the ones that we will be targeting at launch. And if we just zoom further into here, you will find that in Mass General Hospital, which is a large hospital, which has almost more than 40 pancreatic specialists, of those 30 of them we have managed to identify are going to be able to use their test in their clinical diagnostic routine. So they will be the ones that we will focus on at launch. So as you can see, all the commercial readiness and the commercial processes for the launch are in place and ready to go. I would like to give the word to Tom, who will take you through the final clinical validation stages for the launch and all the achievements that have been done in the laboratory in Marlborough in the US. So please, Tom.
Thank you, Laura. Good afternoon. And can we have the next slide?
Yes.
Thank you. So I wanted to very briefly walk you through the last steps towards our clinical launch of our pancreatic cancer test in the US. As I think you mostly know, we spent a substantial amount of time establishing the lab in Marlborough, Massachusetts. And I wanted to say that there is a virtual tour of that lab available on our website if that is of interest to you. Also with kind of a high-speed rendition of how the pancreatic cancer test will be deployed there. We spent a lot of time and care developing those policies. Our ultimate goal, as you can see on the right-hand side of the slide, is College of American Pathologists accreditation. That is really the gold standard for clinical laboratories in the US and to some extent around the world as well. So that's where we ultimately want to be. In order to achieve that accreditation, a lot of things have to be in place, many of which are related directly to the test, but many of the aspects of that aren't. They're the general laboratory, having an established quality management plan that is of high quality and is comprehensive for the laboratory. We spent our time really working extensively on that. We've developed the laboratory procedures and supporting tests that are needed to perform the pancreatic cancer test. We've also validated the FDA-approved instrument we're going to use to measure CA19-9 as part of the pancreatic cancer test. We've done fairly substantial work now in actually developing the LDT in the US. We have assessed its precision. We've assessed sample stability, which is very important for us. Pre-analytical variables or changes that can occur in samples before they reach the laboratory can have a very dramatic and often deleterious impact on the results that are obtained. So evaluating the stability of serum samples in transport has been very important to us. And I'll say a little bit more about that when we talk about the logistics of this assay. We've also been able to evaluate potential interferences, most importantly hemolysis or the lysis of red blood cells, which can occur if a sample is collected improperly, and ascertain what the effects of that are on our assay. So that's a lot of the background work, but it's very important that we have all of this. We've also gained some substantial experience in performing the assay and worked out the SOPs and the training, or SOPs are standard operating procedures, for performing the test and training our technologists to do so. We are now just about ready to begin a blind validation study that is in preparation for our US launch. The regulatory aspects of this first involve application to the state of Massachusetts for a laboratory license. Concordant with that, we'll get a CLIA number from the federal government. And once that initial inspection is complete, we'll be able to perform patient testing. And we anticipate that happening in the next couple of months before we actually begin the sale of IMMray PanCan-d in the US in Q4 of this year. As soon as we've applied to the state, we will also apply for College of American Pathologists accreditation. And that will take about 6 months, we expect, for an inspector to come on site and perform that. I've been a member of the CAP for 30 years or more and have performed many inspections. So I think we're well poised to do that. I've also started laboratories in Massachusetts and so have interacted with that state licensure apparatus. So I really anticipate no problems with either of those. I think we have very strong validation results and we have a very well-developed and well-built clinical laboratory with highly experienced technologists. And next slide, please, Laura.
Yes.
And I wanted to highlight that, my role as medical director, my main role is in patient safety and making sure this test is performed accurately and the patients are informed appropriately of their results and managed in a clinically appropriate manner. But I've also had the opportunity to interact certainly with our staff in Lund, in R&D, and in production, which has been an excellent experience. Also, we've worked a lot with our US team in terms of logistics. Laura had mentioned home draws or remote phlebotomy. That's an extremely important step for us. For a test that's starting off without a large associated clinical laboratory that's already pulling in many patients for blood draws, that was a huge barrier for us. So identifying that, developing partners, and working through that process has been very important for us. Also, having the control over the pre-analytical variables, or the way the sample is collected and handled, is very important for the accuracy of our test. I wanted to highlight one other feature in terms of the logistics of this assay that we've really worked in the laboratory directly with our logistics personnel to develop a means of sample transport that is robust and I think will work for all areas of the United States. That involves a specially designed container that has a cooling engine that can keep samples between 2 and 8 degrees for up to 48 hours. And we've worked to validate the stability of serum samples up to 72 hours in that temperature range. So that collaboration together has been very important. I've also want to say a little bit more about the sales team. I've been involved in their recruitment. These are excellent people, very sophisticated, with direct experience in selling to GI specialists. And I can speak from my own experience in developing laboratories and selling laboratory tests, that having an experienced sales force is absolutely essential. Otherwise, you're spinning your wheels for a long time. And I can say that the individuals we've identified for the different market areas are really of excellent quality and highly sophisticated, and are already experienced in visiting GI offices and speaking with gastroenterologists about complicated tests such as ours. So I think that those things together have really put us in an excellent position to move forward and launch this assay successfully. And the regulatory approval, which is really the last step before we can launch, I think is more a formality than anything else. I really don't see any problems with that. And with that, I think we will stop our presentation and be happy to take your questions.
Thank you, Laura, Linda, and Tom, for a very informative presentation. We're opening up for questions now, and I have several in the queue. The first question is, was the CA19-9 assay used throughout the entire process manufactured in the same way? Was it the same batch?
Yes, it is. Yes, so of course,
okay.
Yes, very important.
Yes, okay, thank you. So the second question is, some pancreatic cysts are called cystic tumors. Research has shown that these pancreatic cystic tumors develop to become pancreatic cancer with the passage of time. The first question to this is, at what point does our test detect the transition from systemic tumor to pancreatic cancer?
And Linda, would you like me to speak to that?
Yes,
Yeah,
yes.
you can take it, Tom.
You can take that.
Okay, so we have fairly limited experience with IPMNs with our assay. High-grade IPMNs, which are ones that are about to transition to invasive cancer, were detectable in our JCO article with fairly good sensitivity. That's an area of active investigation for us and something that we can tend to learn much more about as we move forward. But at this point, our information is limited on that entity.
Okay, so the second follow-up question to that is, cystic tumors are currently monitored annually by MRIs. Could your test be a replacement for an alternative to MRIs?
Again, I think we hope that it will be. That certainly would have a very substantial economic impact in terms of in persuading insurers to pay for our test. So that is an important part of our strategy going forward in terms of adding information to the management of patients. I think it's important to remember that there is no gold standard for the diagnosis of pancreatic cancer. So I think one must, as is specified in NCCN guidelines, take information from all the available modalities and put them together with a number of experts in those fields to try to make decisions about individual patients. I think that our assay will contribute significantly to that. And I think as we deploy the test, our information will grow. And I think our confidence and ability to provide clinical guidance will increase over time.
Great, thank you. The next question is, has the non-PDAC control samples in the verification study been collected at the same clinical sites as the PDAC confirmed blood samples?
Yes, we have non-PDAC controls from several different sites, and we have also PDAC samples from several different sites, and they are also from the same site. So we have a great opportunity to both compare between sites and within sites. So that is very important to see the reproducibility of your analytical performance of each and every marker. And that's something we also have looked into for this commercial test model study.
Great, thanks, Linda. The next question is, do you have any figures for PDAC stages 1 and 2 versus diabetic controls?
The diabetes controls are a part of our symptomatic controls. So these are part of this group. And today we have shown very strong data on diabetes before, and now we are collecting more data on the ongoing studies, and we will have even more data from our prospective trials that are ongoing. PanDIA have, for example, a very large collection ongoing with over 5,000 diabetes. So we are collecting more data specifically on the diabetes as a subgroup within the symptomatic controls.
Great, thank you. Were the results from the commercial test model study obtained after the signature had been fine-tuned and locked, or has the signature been fine-tuned after the study?
No, it was fine-tuned before. So the performance data I've shown today is from a test set where we have built a model with a locked signature.
Great.
Yeah.
That seems to be all the questions at this point. Oh, there's one more coming in. Actually, this is a question on sales forecasts and how many tests we expect to sell in 2021. And I will actually take this question and say that this webinar is not answering those types of questions. If you want to email me, juliesilber@immunovia.com directly, we can answer that question. And I will make another comment out there. Those of you who want to ask questions, please feel free to type those in at this time. It doesn't look like we have any more questions coming in at this time. So I'm going to thank everybody for joining us today and thank our presenters. I think this was a wonderful expansion on all the information we've been sharing. And thank you for joining us today.
Thank you very much for listening. Thank you.
Thank you.
Thank you.
Thank you very much.
Thank you. Bye.
Bye.